Good morning, student. Today I am going to tell you about the codominance. In our last class, we had discussed about incomplete dominance. In incomplete dominance, as if you remember, we have seen that when red colored flower is crossed with white colored flower, that time in a F1, in a progeny, we are able to see the different characteristic, that is pink color flower. So it proves that the blending of characters takes place in case of incomplete dominance, means the dominant character is not completely dominant over the recessive one. Okay, it's about the incomplete dominancy. Okay, you can see here incomplete dominancy in progeny. Clear? But in case of, in second case, if we discuss about the codominance is very interesting process, children, because in case of codominance, the F1 generation resemble with both the parents. You can see here, when white colored cattle crossed with red colored cattle, that time you can see in a F1 generation roan color cattle can be formed. Why is it so? Because both the characters are present separately, means both characters are present in F1 generation. Clear? Both the alleles are present in heterozygote condition. You can see here, capital RR dominant white color cattle, and that small rr mean that red colored cattle. When it crossed with each other, that time roan color cattle will form in F1 generation. Clear? The alleles which do not show dominant recessive relationship and are able to express themselves independently. You can see that white patches are there, red color patches are different, both the colors are visible. Clear? So it proves that this type of allelic form is known as the codominant alleles. Clear? Coat color of these animals are easily expressible about the codominance. Clear? Here you can see that dominant character means white dominant and red recessive variety appearance of all roan offspring. You can see white color is dominant, but red color is also present in roan color variety. Both the colors are visible. So it proves that in F1 generation, roan and red color in 1:2:1. Clear? In F2 generation, in F1 generation all are roan color, but if you cross it again, self crossed also, in F1 F2 generation you can see the ratio in this way. So it proves that the same ratio you have seen in law of dominance also. Clear? But what is the drawback about law of dominance? It always say that dominant characters are visible, but here we are seeing just exception in this law. Clear? That when white colored cattle is crossed with red colored cattle, that time roan colored cattle will form. Okay, the roan colored F2 individual in above cross have both red and white hairs in form of patches. Clear? So here you can see, are you able to see any kind of intermediate color? No, clearly both the colors are visible. Okay, so it's about the codominance in case of cattle. But one more interesting example is given in your NCERT book related with codominance. It's about the blood group, human blood group. Okay, how the human gets the blood group, that is the question. Clear? A good example is different type of red blood cells that determine our ABO blood group. Clear? Do you know that how ABO blood group are controlled by the gene? Clear? Our blood group is controlled by gene which is I. You can see here I is written. I is controlling gene of ABO blood group. Clear? Plasma membrane of RBC. RBC's plasma membrane contain different type of sugar polymers, or we can say that different proteins or antigens. Clear? Which carry antigens? The outer membrane of plasma membrane, clear, and it is controlled by three types of alleles: IA, IB, and third one is I, clear. Alleles IA and IB produce slightly different type of sugars, clear. They both are different from each other, and you know humans are the diploid organism, and each person carry two types of I gene alleles, clear. So IA and IB, as I told you, they both are dominant, and I is small I1 is recessive, clear. So wherever IA IB is present, definitely it show its protein content, clear. One more picture I am just sharing here, you can see on your screen: blood type A or blood type B, clear. When they both crossed with each other, that time the four possibility will come. Means if IA, you can see blood type A, blood type AB, which is co-dominant. Means co-dominant, why is it so? Because both the characters came in case of AB. Yes or no, children? Both the characters can come. Both the characters are present in this second case. Okay. So here it is visible that co-dominant stages there mean if both the characters are present in an individual from their parents, clear, no blending of characteristic, so that time it is only known as the codominance. In third case, blood type B can be formed, and in last case blood type O can be formed. Now again I am in that table, and I want to discuss one thing. Okay, you can see on your screen that whatever the antigen is produced by I allele, that time only this way we can say that particular organism gets the blood group. Okay, easily we can understand by this process that IA and IB are completely dominant over I, clear. So IA and I, small I, you can understand that IA and small I are present only if this condition will present, it proves that A blood group. Okay, if in second condition, if IB, that time which blood group? B blood group. Okay, B blood group is present. So it proves that in third case also, if suppose IA and IB both the conditions present, that time which blood group it is? A B blood group, clear. If only I condition is present, that time definitely it's of O blood group, clear. So through this chart you can understand that allele form parent IA, allele form parent IA, you can see in a first condition. So genotype what will form? IA, definitely A type of blood group. In the same case IB, IA, AB blood group. In third case IA is there. Okay, and in second case small I is there, means no antigen. So that time which type of blood group? A. Second case IA, IB parent 1 and IA parent second, that time AB. Okay, if IB or IB, both the cases, both the parents are having same kind of blood group, that time also B blood group. IBI B blood group, or II small I O blood group, clear. So by this process only we can see that only codominance is present in which case? This case. Okay, where AB, both the parents characteristic comes in progeny. Okay, so IA, this IA come from parent 1, this IB comes from second parent, but characters are not blending. In this case, suppose it's a plasma membrane of RBC, that time A antigen is also present or B antigen is also present, clear. So that is the way we can identify the blood group in humans. Okay, if suppose IA is there or I not, or we can say that IA or small I, that time suppose a plasma membrane, which type of antigen will present here? IA. Okay, I hope that is clear, but if you have any query, any question regarding this, you can send me your question. Thank you, children.